So, yes, I find that this is a hard question to answer, as many of the phase 2 studies are evaluating promising compounds or testing provocative hypotheses that are based on observations we're seeing in clinical practice. So I feel like on the one hand, a number of them have the potential to shift clinical practice in the near term. At the same time, with the approval process, some of them may take longer. So, of the studies that I presented, though, with focusing on those that were evaluating the novel radiopharmaceuticals, probably of those, the ones that will shift clinical practice the earliest, my guess is that the alpha emitter drugs that target PSMA and a somatostatin receptor. And the reason I say that is these are known targets, and we have demonstrated efficacy by targeting these with radiopharmaceuticals with our approved drugs, lutetium-177 PSMA and lutetium-177 dotatate. Therefore, given the differences in how alpha emitters send out their energy and kill cancer cells versus lutetium-177, we know that these targets work well in terms of delivering drug. And so hopefully with alpha emitters, we might increase that efficacy by continuing to target these molecular receptors and delivering the energy with alpha emitters that way. Additionally, there are novel drugs that are targeting, again, PSMA and dotatate to increase their retention time in the tumors and provide potentially even higher tumor targeting and lower background. So one drug that I presented that would fall into this category is copper 64 and copper 67 SAR bis PSMA, which is a tweaked compound that targets PSMA. It's a novel radiopharmaceutical pair in that it uses copper 64 and copper 67, and the initial results showed very high uptake with relatively low background and higher than some of the PSMA agents we already have. So drugs like that are likely to be adopted more rapidly into clinical practice, given that we are familiar with lutetium-177 PSMA and lutetium-177 dotatate already. Additionally, I covered a number of other phase 2 trials that are testing various applications of alpha emitter drugs in combination with other drugs. And so these also show promise, and including lutetium-177 based drugs, some of these are also being tested in combination with other drugs. These may show promise in other tumor types, but it remains to be seen how quickly that will shift clinical practice. But given that these, especially the ones that are looking at lutetium-177 dotatate and lutetium-177 PSMA that are already approved, if any of these trials show efficacy in combination, those will likely move into clinical practice more quickly, such as the use of enzalutamide with lutetium-177 PSMA. That clinical trial was quite positive. Finally, the FAPI-based radiopharmaceuticals, I didn't talk about them so much just because at the time I did the presentation, there were not as many phase 2. Many of them were moving in phase 2 to phase 3, but many FAPI-focused radiopharmaceuticals are out there and being tested in phase 2 and phase 3. And I think that's probably one of the up-and-coming classes that may make it into clinical practice and have a big impact as well. So this is a great question because the classic theranostics paradigm for radiopharmaceuticals, we're seeing that being brought into clinical trial design. So with theranostics, you have a diagnostic, and in the world of radiopharmaceuticals, it's usually a PET or SPECT imaging radiopharmaceutical that you use to image the tumors and see how much uptake there is. And then you can treat it with the same or similar compound, but now labeled with lutetium-177 or an alpha emitter or another therapy isotope. So we're already seeing that paradigm incorporated into clinical trial design, particularly in phase 1. In the phase 2 space, we still are seeing dosimetry, so the use of the SPECT imaging of lutetium-177, for example. We're seeing that used in phase 2 trials to help optimize the dosing strategy or confirm the dosing strategy in phase 2. So I would say that is a novel way of using the theranostics paradigm to hopefully improve our ability to develop compounds that are more effective. Additionally, because we also have data from prior clinical trials with lutetium-177 dotatate and lutetium-177 PSMA showing that PET imaging can be used to select these patients, we are also seeing that paradigm used uniformly almost in phase 2 clinical trials. So nearly all clinical trial designs require patients to undergo some sort of PET imaging or other molecular imaging study to then select those patients with high enough uptake in the tumors to warrant proceeding with treatment. Additionally, we're also seeing incorporation of these molecular imaging studies into clinical trial designs for other treatment modalities, such as immunotherapies that are targeting PSMA. So that's another broader application of using molecular imaging that potentially could improve our ability to develop multiple different treatment modalities, not just radiopharmaceuticals. I think my answer to the last question actually answers this question. We are indeed seeing an improved ability to identify those patients who will respond better to radiopharmaceutical therapies when we use pre-treatment imaging such as PET. So, and we see this in our standard of care approved drugs with lutetium-177 dotatate and lutetium-177 PSMA using dotatate and PSMA PET to select those patients. Because the definition of theranostics is the use of a paired diagnostic and therapy agent, where the diagnostic agent can give specific information about the potential for drug uptake in tumors, I think clinical trials for radiopharmaceuticals are being designed to incorporate molecular imaging. The data that we're obtaining from these clinical trials and the clinical trial designs themselves, I could see them creating a natural path for these, leading to imaging-based biomarkers being approved also as companion diagnostics for theranostics applications.